Yes, so thank you very much, Dr. Kosmidis, for the invitation to come here, and also thank you for having me today, dear colleagues of the Greek Society, and I'm happy to share with you some of our thoughts related to difficult neuroblastoma situations. And what I'd like to do is to have a three-part presentation, basically, and start with some work that we have been doing with the anti-GD2 antibody in the frontline as an introduction, and then develop the idea of what needs to be done eventually when patients that have received this drug relapse. And this is some things that we are constantly working on and try to also structure what we are doing. We are not having a unified protocol at the moment, but this is eventually the goal in the future, that we have a common approach for patients that eventually have a relapse of neuroblastoma. So the survival rate in high-risk neuroblastoma has really improved over decades, and this is shown in this slide here. On the left side you see the Kaplan-Meier curves developing over the era of diagnosis, and what you can basically see is that in the last century, the blue curve shows that the outcome was not very good, and as we move on and develop new treatments, we certainly make a progress. However, the cure rate is not really outstanding yet, and in particular when you look on the other side, which is acute lymphoblastic leukemia, here the development of the prognosis is much more successful, and so we need more novel approaches that are capable of changing the development also for neuroblastoma. So we are advancing, but we have to do more. And in this context, I think it was very important that there were some major results that came out of the European high-risk neuroblastoma study, and one major step forward to improve the outcome is most likely that we have to give a certain consolidation, which is the BUML conditioning regimen. And what is also very important, we have now the introduction of the anti-GD2 therapy to the management of neuroblastoma cases. And in order to show you some very some key information related to that statement, we have just finished analyzing our own experience in this context. I mean, in the past we relied on COG data from the North American group, where RSU has published now also about 8-9 years ago that a combination of anti-GD2 antibody with GM-CSF and interleukin-2 shows a survival benefit at the 2-year event-free survival. And what is nice is that in our own data in the high-risk neuroblastoma trial, when we compare patients that receive anti-GD2 with a time period where this antibody has not been available, we can find very similar 2-year event-free survival dates. They are also at 65 ±2% for the patients that receive chimeric 14/18/CHO compared to the previous era where there has been only standard treatment, where in the maintenance 13-cis-retinoic acid has been given. So that shows we have a clear step forward. However, what you also can see is that the curves are not stable, so they are decreasing. So there is even at 6 years, 7 years, there are patients that still relapse. So that's a problem. But we made progress and we improved the survival by giving this anti-GD2 antibody. Now, a major information was related also to the additional use of cytokines in the context of anti-GD2 therapy. And I told you that in the North American group, the antibody has been used with IL-2 and GM-CSF. And within SIOP-N, we addressed specifically the question, is it necessary to add in interleukin-2 for the antibody treatment? And the rationale to do additional IL-2 treatment is given because IL-2 is a very strong immunostimulatory agent. So you improve the immune system, and the idea was to give that and expect an improved survival. However, we see that the curves are identical, which indicates that the addition of interleukin-2 does not provide an additional benefit. But The event-free survival curves are very good, so just the antibody achieves a survival of 67% here, and so this is exactly the same like here, and it basically shows to us that interleukin-2 is not necessary to achieve an additional effect with anti-GD2 antibody. Very important information, because there's lots of toxicity associated with IL-2, fever, capillary leak, patients are feeling really sick, they have flu-like symptoms, and so it's a big step forward that it's not necessary to use this cytokine in addition with the anti-GD2 antibody. Of course, you know that this antibody has been approved by the European Union in May 2017, and we are now happy that at least in some European countries we don't have a major access problem. Also reimbursement is established in some countries, and this of course needs to be worked on further. In some countries it's not available yet for reimbursement. But the problem is really here, so we still have a big gap. So we want, if you want to be as good as in leukemia, we still have ways to go, and therefore there's novel approaches that we have to work on. We have heard about the option of MIBG, so this needs to be systematically looked at, and eventually we have an opportunity to further improve survival. But anyway, we still have ways to go. So relapse situation is very grim. So we have a rate of 30 to 40% of patients that eventually have relapse after successful maintenance treatment. And what I'd like to share with you are some concepts that we are currently working on related to how to deal with a patient that has relapse of the disease. And this is work that has been done within the German group focusing on relapse strategy, and it's very similar to the European SIOP-N relapse strategy. It basically shows that we need some effective treatment for a reinduction. So when a patient has a relapse, we need to think about what are good strategies to rescue this patient. And at the moment, there are different concepts that we are at the moment working on, and one very interesting strategy is to use also chimeric 14/18 in the context of chemotherapy. And I'd like to show with you some more detailed information why it makes sense to do that. This is not our own data entirely, but there's also data from the North American group, but we also have some initial pilot patients where we use this antibody with N5 and 6 German induction chemotherapy regimen that we can really show that there are some very encouraging response rates when we add this antibody to chemotherapy. So the trick is really to find an effective chemotherapy to reinduce, and then what we then do is to expand the induction treatment with RIST. So this is our German induction protocol. It consists of irinotecan temozolomide. However, the density of the treatment is different. So we give it every 2 weeks, and that is certainly an important aspect. But it's also then asking a randomized question. So one arm of the study receives additional drugs using rapamune and Sprycel. So these are mTOR inhibitors and also tyrosine kinase inhibitors. And the study is still open, but about 3/4 of the patients are recruited. So we expect, hopefully within, let's say, 12 to 15 months, that this study is completed, and we can give an additional idea of whether these additional drugs are important to the success of that rescue treatment. And then there's also in the SIOP and ITCC context the Beacon study, which we are not really following intensively in Germany. So the consolidation strategy, so we have the MIBG therapy here as well. We don't know at the moment in a very structured way, how much you should give it, how to give it, should it be a tandem treatment, what's the correct dose? So these are all questions we are also not clear, and we have to look certainly at the Veritas initiative, how this will move along. And there are German centers that will also participate to the Veritas protocol. My center will participate to it. So it's not clear how exactly to do it, but it's clear that we think that it's important to use MIBG therapy. And then we also know from German data that patients have a benefit when you subject them to a second stem cell procedure. This applies to the autologous transplant. So Frank Berthold and Thorsten Simon have looked at the German GPOH database and identified clearly patients that benefit from a second stem cell procedure after relapse. And what we're also working on at the moment is the concept of haploidentical transplantation. So if a patient comes off the induction and MIBG therapy, we subject them to haploidentical stem cell transplantation. I will share with you the rationale for that and also some initial data in this context. And if you have a patient that is capable of moving all the way through, it also is very clear that such a patient would benefit again from immunotherapy using chimeric 14/18. And at the moment it's still with interleukin-2 because we have a randomized trial open where we have not answered this question whether IL-2 in the relapse setting is necessary, but so we have to wait. But from the frontline data, we think that IL-2 may not be necessary, but this answer is not clearly established at the moment. We also use the minivan trial, so I'd like to share with you the rationale for that, and there are some other studies that we are using. So it's really a structured way through. So when there is a patient, get a biopsy, also very important, because we have in Germany and also I'm sure in Greece there's also pathology laboratories that can identify druggable targets. ALK, for example, is one, but also there are certain kinase pathways that are druggable, and therefore we want to know if such a patient has a gene signature that could be useful to identify small molecule inhibitors that can be introduced in the relapse strategy in order to further improve the outcome for such patients. So this is the overall concept, and what I'd like to do is to show you some basic elements of that concept, and it is organized in maintenance, consolidation, and induction. So I'm going a little bit backwards here. So we start with the maintenance of relapse refractory treatment, then come with the consolidation, and then start with the induction at the end. So what we've been working on for a long time now is the application of the anti-GD2 antibody in relapse refractory setting and change the way how this antibody has been applied. And the reason to do that is because we have a very specific side effect, which is the induction of neuropathic pain. If you give anti-GD2 antibody, the antibody binds to pain fibers and you need IV morphine to manage such a patient. And this is a real problem and it's very uncomfortable. And therefore we developed a long-term infusion schematic where we give the antibody at the same dose but over a longer time period. And we tested this in 53 patients that have been treated in my center, but also started a prospective trial with this question in SIOP-N. The compassionate use program has been done at my center. We enrolled patients between 2009 and 2013, so it has been now completed for 5 years. And we, this is the treatment schematic, so we give the antibody for 10 days continuously, and we also have the cytokines still implemented here because we didn't have the results of the randomization. And here is the 13-cis-retinoic acid. And what we did, we analyzed the toxicity with that treatment, and what we could clearly find is when we start with the standard dose of IV morphine, we observe that the patients require less and less morphine over time, and even the majority of patients did not need any morphine after 5 days. And this is even less the case in the second cycle, third cycle, fourth cycle, and so on. In cycle 6, none of the patients needed any IV morphine. So this indicates really that this type of delivery provides an advantage, makes it much more tolerable, and what we can also do is we actually can send the patients home after a few days. So they get the antibody in a pump, and when they feel good, you don't need any IV morphine, we discharge them and they go home and get their treatment there. They come, of course, on a regular basis for checkups, but the treatment is much more tolerable in this way of application. This not only relates to pain, it also relates to other side effects, such as capillary leak syndrome, for example. It's much less when we use it as a long-term infusion. Fever is less frequent, and all the other side effects are also less frequent, indicating that this is the right way to give this drug, because it is also inducing responses. So, okay, when you change the delivery method, you want to make sure that you don't lose the efficacy of the treatment, but that's not the case. So in relapse refractory patients with measurable disease, we can find a 40.5% response rate. I mean, in the past, we all believed antibody can only work in minimal residual disease situations, where you only have minimal cells somewhere sitting and you are capable of preventing a relapse from that. But that's not true. We see patients that have MRI positive disease, that have MIBG positive disease, that they actually respond to this type of treatment. And this is the response rate we observed in this cohort. Now, this is not a randomized study, so what we have to do when we look at the overall survival, we have to compare with historic controls, and here we see a dramatic improvement. What we used here is the Garaventa cohort collected at the Gaslini Children's Hospital, and when we do a matched pair analysis, we see that we have a very clear signal of a survival improvement when we use this antibody after relapse or refractory disease. Now, this is, of course, a single-center experience. You always want to make sure that this holds up when you do a multi-center clinical trial, and therefore we launched the LTI study. And the LTI study has two phases. One phase is very mature. It's the single-arm phase, where we give the antibody in combination with IL-2, and then we started a randomization from 2014 to 2017, where we also ask the IL-2 question. This part is not mature enough to be reported, so we still have to wait for data to come in. But what I can share with you is data related to the single-arm phase, where we use the antibody combined with IL-2 in all patients. But I think the information here is very important because it also shows the confirmation of the compassionate use data that I've shown you previously. So this shows again the treatment. It's exactly the same like in the compassionate use cohort, antibody as continuous infusion, but combined with interleukin-2 and also 13-cis-retinoic acid. So the main information is shown here. So when we look at the response rate, we find at the mid of the treatment, which is after 2 cycles, a response rate of 50%. And this remains at 43% after 5 cycles. So it very nicely shows that response rate is exactly the same like in the compassionate use program. This is the event-free and overall survival. So also here we can have only a single-arm observation. So these curves don't mean by itself very much, but what we have done is we try to analyze in this cohort whether the antibody mechanism is really holding up. So we looked at certain polymorphisms of the Fc gamma receptor in order to see if we can identify patients that have a better response compared to patients that have a lower response. And we did this related to the Fc gamma receptor polymorphism. So every human is equipped with a high, intermediate, and low affinity of these Fc receptors. So the NK cells use this receptor to recognize the antibody on the cell surface. And we can group our patients according to either a low affinity group, so these are patients that have a low affinity of their Fc receptor, and compare the outcome to patients that have a high affinity of their Fc receptor. And when we apply this information, we can show that indeed patients that have a high affinity Fc gamma receptor polymorphism show an improved event-free survival and overall survival compared to patients that have a low affinity Fc gamma receptor polymorphism. This is important information because we then can eventually work in the future on a biomarker. So and then at the moment we don't have a good idea what to do with these patients, but eventually if there is a good solution, one could think about different treatments for patients that have a low affinity Fc gamma receptor polymorphism. So this is just using the antibody by itself, but what we also do is we want to work on novel combinations, and you are all aware of that the Nobel Prize this year has been given to a very crucial observation, which is that you can have a very, very active cancer therapy by the inhibition of negative immune regulation. And there are two landmark observations that support that, which is given by Jim Allison and CTLA4 molecules, and also by Tasuku Honyo by the PD-1, PD-L1 pathway. And this discovery led to an explosion, basically, of the use of antibodies that block these checkpoints, but mostly in the field of adult oncology, because here we have, in some indications like melanoma, very immunogenic tumors. So these malignancies by themselves already are characterized by a strong infiltration of the immune system. And we do not have that in pediatric cancer. Pediatric cancer usually are cold tumors. Cold meaning that they don't induce a very strong immune infiltration. However, when we add an immune intervention in pediatric oncology, the scenario changes. So you can turn a cold tumor into a hot tumor, and we have analyzed this in neuroblastoma very systematically. So when we have a neuroblastoma cell line here, and we measure the PD-1 expression on that, the PD-L1 expression on that tumor, you see basically it's completely negative. There's no PD-L1 expression whatsoever. However, when you add anti-GD2 antibody and the NK cells or any immune cells, and you generate this situation where you have, like in the patient, an immune activation and also a killing of the neuroblastoma cell, what then happens is that the PD-L1 signal on the neuroblastoma is very strongly upregulated. And I think that is the big problem in immune oncology trials at the moment, that to make a decision of whether a patient will benefit from a PD-1 checkpoint inhibition is a baseline biopsy of a non-treated patient. And I think this is the wrong information. What is clear here, that if you give an anti-GD2 antibody and you start such a treatment, the checkpoint is upregulated, and for the tumor it makes a lot of sense, because this tumor is then protected from immune attack. And therefore you would then develop a completely novel way of looking at this problem. Now, what is interesting is that this effect is highly specific. So when we block the binding of the antibody to GD2 with an anti-idiotypic antibody, which is an antibody that prevents recognition of GD2, the upregulation of PD-1 is completely abrogated. Now, we analyzed also the situation when we add interleukin-2 to this, or we change the construct. So when we add humanized IL-2 cytokine to this effect is even more pronounced. So the upregulation of the PD-L1 checkpoint is even stronger. And this is the bar graphs to show that. So the fact that we have a strong upregulation of this checkpoint opens a new idea to combine these treatments. And this is what we've done. So when we look at an ADCC assay and we block PD-1 in an immune silent tumor, we can upregulate the killing. And when we translate this also in a mouse experiment, so when we inject neuroblastoma cells and we treat with anti-GD2 antibody in combination with anti-PD-1, we clearly see a synergistic effect. So the primary tumors are growing slow, but they're not growing at all. So the size of the primary tumor is lowest in the combined group. The survival is also the longest when you give the anti-PD-1 antibody combined with chimeric 14/18. And this is basically the scientific rationale to do this Minivan trial, because here we really have a combination of chimeric 14/18 with the nivolumab as a core part of the immune intervention following the MIBG therapy. The MIBG MIBG therapy here is a component that is also improving the immune checkpoint situation, but anyway, the core part is the combined anti-GD2 immunotherapy with anti-PD-1, and here are the primary investigators. It's led by Juliet Gray in Southampton, together with Mark Gaze, who is doing the MIBG part, and my center and Paul Sundell's center in medicine are participating to that study. So I have to watch the time a little bit. I don't know how much time I do have left. I'm okay. Yeah. So, okay, you just give me a warning when I have to stop. So now we're leaving the maintenance phase, but move into the consolidation. And I told you that the intervention with a second stem cell procedure seems to be important in patients that have a relapsed disease. But what we are more and more focusing on is to change from the autologous setting to the haploidentical stem cell transplant setting. And the reason to do that is because we have a certain immunological reasoning behind that, which I'd like to share with you. Another reason is that we have a very motivated donor usually available, which is the father or the mother of the child. And therefore it is completely straightforward to then establish a transplant procedure based on such a situation. And the reason behind that is that after transplantation of stem cells that were gained from father or mother, we usually have a very strong increase of natural killer cells. So there's a very large number of these cells in the graft itself, and this is the first cell population that increases after the transplantation, and therefore it is a very straightforward idea to use then a monoclonal antibody to direct these NK cells against remaining tumor that is in the body of the child. This is the conditioning regimen that we are using. So we also have an MIBG therapy component here, 3 to 4 weeks before the conditioning is starting. And this is containing a T-cell directed immunotherapy using ATG to get rid of the T-cells of the child in order to avoid the rejection. Then we have also fludarabine, tiotepa, and melphalan, which are at least the melphalan and the tiotepa drugs that are active also against neuroblastoma. And then the patient receives the stem cells that are clearly T-cell depleted. So we have a very good T-cell depletion. Otherwise, you get a strong graft versus host disease. And at the moment, we are using a CD3, CD19 technology, but there will be a change in the future using the beta alpha depletion, which has certain advantages eventually in the future. So the whole package looks like this. So the transplantation is done, and then after 60 days of the transplantation, we come in with the anti-GD2 antibody, which is given over at least 6 cycles. Some patients also receive 9 cycles. And the first 3 cycles are given without interleukin-2, and the last cycles we have a small amount of interleukin-2 implemented here in order to see if there is an advantage in terms of additional immune stimulation with this cytokine. Now, what I'd like to share with you are some initial results in this context. So first of all, we have the treatment here related to the response rate. So there's over 60 patients that are included in that trial, which is led by Peter Lang in Tübingen, and we as a site are contributing to these patients. And this graph is maybe a little bit complicated, but if I guide you through it, it's not too complicated after all. So when a patient goes into the haplotransplantation and has a complete response, 90% have a complete response after the anti-GD2 antibody therapy. However, there's a few patients that also have progressive disease. One has to be aware of that. So it's in the range of 10% that come in CR, but then have a progression after the whole procedure. So, however, the encouraging thing is that there are patients that have a remaining disease before the intervention, like partial response or mixed response or stable disease, we have then patients that to an amount of 41% turn into a complete response. And some have a partial response. So the total amount of patients that have a complete response after that intervention with measurable disease prior to the intervention is at the range of 41%, which is, I think, very good. So with the event-free survival, also here, it's not a controlled trial, so one has to use historical controls, but these are very good historical controls because these are controls that only received the haploidentical transplantation with n equals 26. And these are the patients that received the haplotransplant with chimeric 14/18. And we see a clear difference in the event-free survival with a p-value of 0.001%. So this is a very strong message that the chimeric 14/18 following haplotransplantation is adding to event-free survival in these patients. So the main conclusion from that trial is, first of all, that the haplotransplantation and anti-GD2 therapy is feasible. There's no major toxicity. So the big worry was that there will be graft versus host disease. So if you do a haplo and you come in with an immune intervention, then you will see very strong graft versus host problematic. But it's not the case. So in fact, there is a small percentage that have grade 2 to grade 3 GVHD that has been treated successfully. So no patient died of that intervention. So the response rate is very encouraging. Overall, an event-free survival is very encouraging. And major influence factors on EFS is the remission status prior to the intervention. So the best scenario is to achieve a complete response before you actually get the treatment. However, patients that have remaining disease still benefit and have a response rate after that intervention. Okay, so this is the consolation. So we are very interested on the haploidentical transplantation perspective, and we also working on eventually doing a randomized study to really pinpoint this haplo effect, but this is a little bit more in the future at the moment. So we hope that we can get such a trial designed and open. So if I have a little bit more time, maybe 5 minutes, I would like to share with you the rationale to combine anti-GD2 antibody also with the chemotherapy. And this is work that I think is very exciting, and it has been started with a trial in North America, where the anti-GD2 antibody was combined with irinotecan temozolomide in patients that had recurring or progressing neuroblastoma. And this is a randomized study, okay? So these patients received in one arm the anti-GD2 antibody with the chemotherapy, and in the other arm they received temsirolimus in combination with irinotecan temozolomide. And the surprising thing was that the patients that received the antibody in combination with the chemotherapy had a dramatic response of 53%. I mean, that was outstanding. So they prematurely stopped the treatment and stopped the randomization and had an expansion cohort. And this expansion cohort has been reported this year at ASCO and also at the ANR meeting. And the expansion basically shows that the response rate is not 53%, but more 40%. But still, it's a very good response rate, and encourages the group to move on forward to use that combination. There's other signals that the immunotherapy is working together with chemotherapy. So there's a big group at St. Jude's where a variant of anti-GD2 antibody with the deletion of the complement binding site is given and in combination with induction chemotherapy, and also here a very good response rate has been observed. There's no long-term follow-up at the moment, but the response rate certainly is very promising. So this is why we also started to work on such an approach. So we selected our German induction chemotherapy cycles in order to get a first idea of how such a treatment would be actually feasible at all, because you see that these chemotherapy elements are quite complex, and we wanted to explore if you actually can do such a combination at all. And we used chimeric 14/18 also at a lower cumulative dose at 50 milligrams per meter squared, so not 100. And the reason for that is that these chemotherapy cycles are usually denser. So you give that every 3 weeks, and in the maintenance we give it every 5 weeks. So therefore we go down in the dose of the anti-GD2 antibody. And what we've done, we piloted patients, a total of 16 patients between the Charité, my center, and also Vienna, where we had relapsed patients that had a certain baseline characteristic you show here. They are, except for one, all relapsed stage 4 or stage M neuroblastoma. And at the inclusion, all of them failed at least one second-line chemotherapy. So they are this really bad population. Time from diagnosis to relapse is also relatively short. It's 1.8 years. And the type of relapse is mostly disseminated. So the previous therapies are shown here of that cohort. So most of them received previous N5 and 6 chemotherapy. As I said, all patients failed at least one second-line therapy. The way how we did that is that we planned a total of 4 cycles, and we evaluated after 2 cycles. And it was an alternating regimen. A total of 4 cycles were applied. We also checked grade 3, grade 4 toxicities. And then we determined the response with the usual methods, MIBG scan, CT MRI, and the results were that a total of 43 cycles were applied. And it was feasible. There was no major toxicity that we observed that were not expected. And we could follow-up is very early, so we completed that by the end of last year. And we are in the process of collecting the first event-free and overall survival data. Of course, most likely many of these patients will have a third, second, or fourth relapse, and so one has to be honest that this is not a solution to the problem, and it's really only to get a first idea whether you can give such a treatment in combination. But at least the signal we are getting is that it's very, very active. And therefore we want to also look at that in a more systematic way, and we are planning to implement the GPOH induction regimen in the future high-risk neuroblastoma protocol. And in order to do that, we want to first get a very good idea of the feasibility in this context, and therefore we designed this pilot studies, which is a thing that has been emerged already to a very late stage. So in fact we have a finalized protocol, and we hope that we can get all the regulatory stuff done within the next half year, and then also have a protocol open to address this very exciting question. So at this stage I'd like to summarize. So we really start to work on a very structured way how to handle relapsed patients, get them all a biopsy, so get good biological information, but then also work through the steps of induction, consolidation, and maintenance. We think that the majority of patients that will benefit from chimeric 14/18 also in the relapsed scenario, and early clinical data also suggest activity in combination with chemotherapy. So with this said, I'd like to stop and show you some faces of a very small but dedicated team in Greifswald working on neuroblastoma. but also, of course, there's lots of people that we work together with, in particular the SIOP-N group, and also Vienna as a center is a very important partner in this cooperation. And this gentleman is also very important because it's Ralf Reisfeld. He's the guy who actually made chimeric 1418. And so we relied on his work together also with Stephen Gillis, who is the molecular biologist who made the humanization of the antibody. That we have now, I think one of the very first time a drug that has been approved for neuroblastoma. So it happened in history of drug development 4 times so far that a drug has been developed for pediatric cancer. Usually it's the other way around. It's developed for adult cancer and then it is used in pediatrics. And this is one example where it has been done the other way around. So thank you very much for your attention, and I'm happy to take questions.